Sorry, I'm on mute, but also saying no problem.
Thank you.
Okay, well, thanks for the opportunity. I know a number of people on this group, and I am a little intimidated by the level of understanding of all of this. So I will scoot through what I've kind of been asked to present, which is an ecosystem services view, less the biogeochemistry, which Ashley will be touching on. But it's as a reasonably small group. If anybody has questions, wants me to expand or skip over something that's completely known to the entire group, I've had a little difficulty trying to figure out just what level to pitch it at, and I know that there's a reasonable level of understanding of all of this material within the group. So feedback welcome as I go. Yep, here we go. So these usually start with what have we lost, and I was thinking about that and looking back at this Zuermgassen et al. 2012 publication, and struck me that was an interesting one to lead out with, and largely because of that very odd blue patch in Florida. One of the things that Lena Zuermgassen found when she was having a look at a detailed look at the US coast in terms of what the changes in both biomass and also extent of oyster reef was, is that the measures that were taken in the early 1900s, I think this area was in about the 1920s when things were being fairly heavily fished, had actually recovered to 2010 when the last information was available, and we know what that's looking like now. So the top line summary there was there's been a 64% decline in the spatial extent of reefs around the US coast to 2010, an 88% decline in biomass, and we know what the trajectory has been in the last decade since 2010. And unfortunately, those numbers will have changed quite a bit since then. So it did strike me as I was looking through some of that historical ecology type information. But that usually leads to the ecosystem services conversation. Why put them back? And that's because of what we gain from putting those habitats back. I won't go into the cultural services to any great extent. Aquaculture and fisheries, I think, are reasonably self-explanatory. The benefit to most of these systems of having a robust supply of larvae, a protected breeding population that actually contributes to the larval supply, is fairly self-evident. Particularly in the Apalachicola case in the last decade that we've all been watching. So coastal protection I'll touch on briefly, water quality without the denitrification component, which Ashley will be talking to, and fish production. So the shoreline protection, coastal defence, is very site specific. It actually becomes an engineering kind of consideration in my mind. So it is a very location specific consideration. You really need to understand the wave environment, the storm environment, topography and currents. So it becomes a coastal engineering in its detail. But one of the things that I've learned from looking at this, and I therefore like to pass on because it might not be as immediately apparent, is that there are two components to the shoreline protection from habitats. And this schematic is just trying to demonstrate that, with habitats you actually reduce that wave height, therefore reduce the storm surge and run-up flooding caused by those waves, remove your habitat, and there's nothing to stop those waves. But how the habitat operates, this oyster reef we've got in the top schematic here is actually acting as a submerged breakwater. It could be anything, it wouldn't matter at all. If sites are suitable for oyster restoration, then you can actually build a submerged breakwater in a way that supports oysters and therefore the other ecosystem services. And that's pretty straightforward. The one that's not nearly as obvious, and I've learned a lot about, is the friction coefficient. When you start thinking about a seagrass bed or a salt marsh, it might be more obvious. You know, waves touch the bottom at 7 times the wavelength, so they're connecting with the bottom for a long way out. And there's a big difference between a wave rolling over a hard, smooth surface and, you know, rolling over a seagrass bed or an oyster reef, which acts like a sponge. And that friction coefficient that you're creating really draws the energy out of a wave from underneath. So that's really the salient comment that I would add on the way through, is these are very much engineering, coastal engineering considerations, and they're site specific, but that friction coefficient is a far more important component of that engineering than really I had appreciated until I started diving into this. So just passing that along. Water clarity has been measured for a lot of species around the world. The lab measures are dead easy to do, clearance rates or filtration rates, not quite being the same thing. More informative, I think, are some of the field measures. And these have been, Ray Grizzle's already been mentioned this morning. This is work that he's been doing for about 15 years now, so this is fairly well established, although not that easy to actually implement in the field. Again, you need a fairly tame reef that you know a reasonable amount about to be able to have a look at what the impact of the filtration from a reef in the field actually is. But some of the graphs of cestin concentration are fairly compelling. And I should actually point out, I guess, on the way through that, you know, this photo represents a couple of fluorometers just measuring, you know, cestin particle concentration underwater. Between those two is an oyster reef, reasonably well documented in terms of density and size frequency. So you know what the filtration capacity is, and as water flows across that once, you can actually measure the decrease in cestin particle concentration. The other way of looking at water quality, which is really just being developed, I don't have images of it, the NOAA Chesapeake Bay office has started to look at some of the big projects, the Harris Creek project particularly, and have found that satellite images are starting to give a reasonable measure of changes in water clarity around a water reef. I mean, any of us have jumped into the ocean, and I expect it's everybody, with a mask and snorkel on. You know, that twinkling of light on the bottom of the ocean as you're swimming, caused by any ripple on the surface, makes measuring light penetration through water a really noisy exercise. That twinkling, refraction, diffraction of light at the surface means that there's a huge noise environment in which you're trying to measure changes in water clarity. And it makes it pretty tricky. We've certainly tried. It becomes a big data exercise. But what the Chesapeake Bay office are finding is that getting water clarity measures from satellite actually evens out all of that noise that you get on a small scale. So that's one to keep an eye on. EPA are actually looking at that at places on Cape Cod as well. And I hope we've all seen these kind of demonstrations of what that filtration does in terms of regulating cestin concentration and general water quality. You know, with high levels of oyster biomass, you actually have water being filtered many times within the residence time of an estuary, depending on what that residence time is. And there's just 3 with varying residence times here that we've documented. So, you know, y-axis is number of days that it takes to filter the entire volume of an estuary, residence time on the y-axis, and as you change the number of, or the biomass of your filter feeding organisms in there. Certainly the amount of work they're doing for your estuary clarity changes radically. One of the things that isn't really talked about that much in the US is biodiversity and biomass. This is actually the focus of conservation in a lot of countries, well, actually not a lot, but some countries around the world. It's not talked about much in terms of the US, but it is a driver. And this, there are tables like this in the literature all over the place. This is one I've grabbed. I think this is from Lisa Kellogg. Unrestored, bottom, abundance, biomass, and just running your eye down those lists of numbers is all that's required. And compare those to restored oyster habitat. And it's pretty obvious that there's at least an order of magnitude change. And it's that abundance and biomass of particularly forage species that drives one of the ecosystem services that's becoming really compelling, which is fish production. So oysters, as they're filtering, are collecting up all of the nutrient containing particles in the water column and feeding them into that matrix that's the oyster reef. And those biodeposits produced by oysters are in a form that is available, accessible to all of these invertebrates. The abundance and biomass explodes, as does the, and they constitute forage species, particularly for small fish. So this is work that Lena Zwirmgussen and a team published in 2016. It's the fish production. I really hope kind of everybody on this call is at least familiar with this, so I'll run through it fairly quickly. But this comes from a meta-analysis looking at studies of fish abundance around the Gulf and Atlantic coasts. It was actually the West Coast as well, but there wasn't enough information available. And the requirements of the data were very specific. So what was required was studies that measured abundance as a density. So we're looking for an area-based measure of density, and particularly including juvenile species. And the 0+ year class of any fish species is the hardest to get a handle on, so you can appreciate that those studies are relatively limited. There were 31 found across the Gulf and Atlantic, pretty evenly split, I think 15 in the Gulf, 16 on the Atlantic coast. Those studies needed to have either more than one site or more than 1 year, so that there was evidence of consistently enhanced species. And the operation then is to look at the juveniles, in most cases the 0+ year class, get a density on habitat and a density off habitat on a control bottom, what would have been there if the reef hadn't been restored or didn't exist. Subtract the number of juveniles on your control from your oyster reef, so that what you are dealing with there are the additional fish that are associated with an oyster reef. And there are assumptions made, but the assumption is fairly straightforward, is that those are additional fish that are there because of increased survival of those very small 0+ year class because of their habitat. You then simply apply to that additional group of 0+ year class cohort all of the species and size specific growth and mortality schedules from the fishery stock assessment data to come up with lifetime production from those additional juveniles. There are assumptions. I've already talked about one being that what we're measuring is a change in survival of that juvenile or 0+ cohort, and I think that's pretty straightforward. The other one that's worth remembering as we start applying and extrapolating these information is that it assumes habitat is limiting. In most places around the world at this point, that's a very safe assumption. If we start modelling out what happens when we put back large areas of oyster reef, then the oyster reef may not be limiting anymore. There may be more reef than there are recruits looking for a suitable home, and taking advantage of that, what I call a nice secure house with a well-stocked fridge, is what an oyster reef turns into from the perspective of a juvenile fish. So this is what happens when that habitat is limiting. And we've seen some of the graphs that come from this. I hope this is for the Atlantic coast. That y-axis is most easily read in tonnes per hectare, hectare being 2.2 acres, and that's about 3 tonnes per hectare per year from the Atlantic coast. And that production actually asymptotes reasonably quickly within about 5 years. And the species list that is benefiting from those oyster reefs, or we have identified as benefiting from those oyster reefs, is relatively impressive. Gulf of Mexico even more so, up around 5 tonnes per hectare per year, and again some fairly important species. So this fish production is really driving quite a lot of interest around the world, not just the US, in restoring some of these habitats. And it's kind of something that we're trying still, and I know there are people on this call that are involved in this, trying to introduce more formally into the fisheries management kind of framework and get away from the traditional view of fisheries management, which is purely looking at how much you can extract and what limits need to be put on extraction so that you maintain sufficient kind of recruitment to maintain a stock. And introducing habitat into that argument is simply acknowledging that if habitat becomes a bottleneck, then you can really change the survival of these recruits entering that stock. And by putting that habitat back, you're supplying a lot more fish. And this can be quantified and actually used in stock assessment work. So what this has changed over the last few years is really the big part of this is separating attraction from production. It might seem odd, but this argument has raged for as long as I've been involved in fisheries management, and then a lot longer. If you put out structure, a lot of fish turn up. How many of those fish were actually produced because of that structure, and how many of them were just attracted in from elsewhere and is not a net benefit to the population? In fact, probably net negative because they're easy to catch. So this is actually the first example we have globally that I'm aware of anyway, of nailing down a method for actually looking at production, and it's that change in survival of the juvenile or 0+ cohort that we're looking at. And what that gives us is that mechanism for getting into the habitat interaction with fish production and fisheries management, which is pretty exciting. Worth pointing out as we're talking about it, I know this is a shellfish group, but it's not just shellfish. There's a bunch of these critical coastal ecosystems, and we have struggled to look at those habitats individually. But I think where this field has got to go, and hopefully in the relatively near future, is looking at mosaics of these habitats, now that we're starting to figure out how to account for the values of some of these different habitats. We need to be putting them back into the matrix that really is the ecosystem. And these are very preliminary data. These are not published yet, but this is just a quick comparison of some of the work that's going into looking at fish production from salt marsh on the right, seagrass in the centre, and comparing that to oyster reef. So you can see that these critical coastal habitats really do play a pretty outsized role in fish production along the coast. This again is one that I'm kind of assuming the group have seen or at least know of. We've put together an oyster calculator. It's available online. And what this is designed to do really is account for the oyster properties and physical parameters of a bay or estuary, and then give the capacity to look at what changing the oyster biomass would do to your filtration and to your fish production by species, coming directly from those graphs we've just seen. And I know I'm one of a group of people that have been trying to add a third column to this decision support tool, and that third column would be tons of nitrogen processed, and what would changing the oyster habitat, the biomass of oysters in the system, do to the tons of nitrogen processed. And that's something that Ashley's been involved in and will probably touch on. But if anybody's not familiar with this, happy to open that up more. I'm assuming that people at least know of its existence. So that brings us into more the economic impacts. Stimulus is starting to be talked about again, actually, so this becomes more relevant at the moment. But we have had some experience with the Obama era stimulus work, and really figuring out that some of this natural infrastructure work is a great job creator. It also has economic impacts through some of these ecosystem services, which is another area that we need to concentrate on more, but there are good examples. This is one from Texas, Half Moon Reef, Matagorda Bay, and really it's this big impact on the recreational fishing industry, which has not been a great driver in the US to date, but I think it's a big opportunity, because the recreational fishing industry is a real economic driver, particularly in the Gulf. So any of the Florida bays and estuaries, I think, would have similar kinds of results to Matagorda Bay in terms of rec fishing. There's another study along these lines, which I haven't presented, from South Australia for reference. But the other one I just wanted to finish on is Chesapeake Bay. And again, I'm assuming you're all familiar with the 10 tributaries work in Chesapeake Bay, and Harris Creek being the last or the first of those to be finished, and a little bit of habitat, a little bit of history from that. So the important point here, I think, is that from that Chesapeake Bay work, there's been enough time to get a 3-year monitoring completed. That was finished in 2018, 142 hectares, I think, what's that, 350 odd acres. A huge amount of substrate, a lot of oysters put in, but this $53 million price tag is the one that gets interesting, because that's a very expensive project. But what came out of this monitoring was an economic analysis, to have a look at the value, particularly of the fish produced from that restoration work. And the results are actually pretty impressive. The percentage increases of some of the important species in Chesapeake Bay, being blue crab and white perch, are impressive. And what's really interesting is that the kind of flow-on economic benefit of that increased fish production from Harris Creek alone is nearly $23 million a year. And it's these economic studies that quantify the value of some of our ecosystem services that I think we need to be concentrating on, because $53 million is a really expensive project until you realise that you're getting $23 million a year, year in, year out, return on that investment. And that suddenly starts to make it look like a pretty attractive investment. So for my time, that's a pretty quick summary of the ecosystem services and some of the benefits from my view. Happy to take questions on that, if anybody has questions.
Hey, Boze, this is Eric. Sorry, I hear some feedback, but how specific is the calculator that you mentioned for Galveston Bay? It's very, I know I've seen it, but I've never tried to use it. Just wondered your thoughts on that.
Okay, sorry. that was a screenshot that I had available. So I grabbed Galveston Bay. We have populated that website with all of the bay-specific data that were available in 2017, I think it was. And they include estuary volume, the flushing rate, so the exchange rate for the residence time for an estuary, and any current, at that point, oyster habitat information that was available, any state kind of surveys of habitat. So you can pull up any bay or estuary around the coast. Most of them have all of that data. If it wasn't available for a bay or estuary, it just simply doesn't appear, and you need to make some assumptions or find a more recent study to add those data in. So that gives you kind of a pretty free access around the Gulf and Atlantic coast for bays and estuaries. And the other thing, if you're diving into it, that I didn't point out there, is that there's a slider bar on each of the fish production measures that come out as a result, and they're set average fish production from the collective meta-analysis that was done. If you know that the species doesn't occur in your bay or estuary, you can zero it out. If you know they are a particularly common species in a bay or estuary and you get a lot more of them where you're working than the bay next door, you can actually take that up to the upper 95% confidence level of the estimate, that mean that was added in. So all of the parameters there are interactive, and you can apply local knowledge to what comes out of it.
So hi, Boze. I have a question. This is Laura Gesselbrock. Great presentation on important work there. Two questions. First one is, given the return on investment there, I would imagine the state agencies and other funders involved would be falling over each other to get more of this reef in the system. What are the plans for the Chesapeake Bay area to put more of these reefs in the system? And then my second question is about the reefs themselves that were restored. How high was the mulch material piled up, if you happen to know that? Thank you.
Okay, so yeah, look, there's a lot of information. The Chesapeake Bay group, and it's a real partnership of pretty much all of the conservation groups, plus the Army Corps of Engineers, NOAA obviously, that work in Chesapeake Bay. So it's a really constructive collaboration, and it needs to be for the scale they're working at. But they have put a lot of thought, and if you're not familiar with, I could send some documentation. It's worth reading. A lot of thought into how to define what area of reef you should be targeting to apply your definitions of what restored looks like. So how do you come up with a total area of a tributary, a bay? The work was kicked off, 2011, the executive order from the Obama administration basically said we need to put a lot of this stuff back. There was no money behind it. There was a governor's coalition of all of the states that influenced Chesapeake in 2014, I think, put some funding behind it. That work really started in 2015, and what they've winnowed down is 10 tributaries as a plan, 5 in Maryland, 5 in Virginia. The first to be finished is Harris Creek at about 350 acres of reef. I think the Piankatank in Virginia will be the biggest at about 580. And the rationale for what those areas represent is based on historic area and a percentage of that historic area. I forget the actual details. Their definition of success after 3 years is, variable age classes on the reef, and they're targeting a minimum density and biomass, as it turns out, of 50 oysters per square metre on that reef. The depth of reef, so Harris Creek put in over 200,000 cubic metres of substrate. They were looking for around 30 centimetres of substrate. Obviously that varies a bit due to deployment, but they've got 98% success, I think, for Harris Creek on their restoration efforts. And these are ongoing. There's a couple of tributaries in Maryland where they are still going through that process of, calculating the area that would need to be restored to meet their success metrics. And restoration hasn't actually begun, but their target is 2025.
So is the funding primarily coming from the state now, or Army Corps, or who's funding the subsequent creeks?
The coalition that's putting this work together includes TNC, Oyster Recovery Partnership, Chesapeake Bay Foundation, the Army Corps, NOAA's Chesapeake Bay Office, and anybody who can find money to put towards a project in the tributary they're focused on adds it to the kitty. That said, without the Army Corps, they would not be at the scale they are now. So NOAA and the Army Corps have access to more funding than the private philanthropy and other avenues that we've been able to get money from. So they have been pretty key.
Thank you.
Any other questions for Boze? Thank you.
Hi, Boze. This is Steve John at FDEP. So you mentioned obviously the really large scale of the Chesapeake Bay example. Do you know, like, obviously?
Oops, you may have cut out at the critical point there. Sorry, I didn't hear the last bit.
Go ahead, sir.
Is this better?
Yes, I could hear that.
Okay, sorry about that. My question is the return on investment a linear relationship between the scale of a project and the economic benefit associated?
Yeah, good question. And the short answer is we don't really know. Definitely, the bigger the project, the easier it is to measure the impact separate from noise and background kind of variation. So you're more confident in your monitoring outcomes at a large scale than a small experimental scale. But there is presumably a relationship between that oyster reef quality, habitat quality, and some of these ecosystem services. We're still at a point of being pretty coarse about service or no service, habitat or no habitat, and it would be wonderful to be able to pull that apart a bit and start having a look at the relationship between level of service and habitat quality. We don't have a lot of detail on that at this point.
All right, thank you.
Hi, Boze. This is Ed Camp at the University of Florida. I had a quick question for you about the attraction and production. And I guess my question comes from the perspective of someone who does a lot of quantitative fisheries ecology work. It seems like the baseline assumption that you're making with the oyster calculator is that fish that are captured in oyster habitat are produced by the oyster habitat as opposed to attracted by it. So I'm having a tough time understanding how this is actually separating attraction from production. It seems like it's simply assuming production.
Okay, so one of the features to focus on is the 0+ year class component of that measure. So really what it's assuming is that there are larvae, fish larvae available to recruit uniformly around a bay or estuary, and that the habitat has an impact on the survival post recruitment. And that's not something that, you know, we're looking at the adult stage. There's been quite a lot of thought put into whether we can actually quantify your production for, or increase growth, increase production for larger year classes foraging on these productive oyster reefs. We haven't got there. So all we're doing at that point is looking at that 0+ year class. And yes, there is an assumption which is violated in other parts of the world. I don't think so much in the US, that there's been recruitment overfishing and there simply aren't recruits available to recruit onto the habitat, whether you put it back or not. I've run into that in Hong Kong. I think in around the US, we're not at that stage, and there are still recruits looking for habitat to settle onto in you know, from the species that we've got there. So it's a matter of thinking through that 0+ year class, how far they're likely to travel, whether they are accumulating on that oyster reef from a distant source, or whether that distant source has its own population dynamics with respect to a 0+ year class. I think, given that I'm not actually an author on that paper, we should be asking Lena that question, but I hope I've done an honest job.
Other questions for Boze? Thank you so much. That was very helpful. I've used the Oyster calculator myself, and you can tell there's a lot of thought that has gone into it. So we'll send some of these links out. I see someone has shared the Oyster calculator link in the chat box. And Anne, thank you for posting some of the references that Boze mentioned in the box already.
She's hard to keep up with, this girl.
Yeah. Boze, would you be willing to share your presentation as well? Can we share that?
Yeah, certainly.
Excellent, thank you. Okay. So Ashley Smyth with the University of Florida. Would you like to present on oysters and nutrient pollution, a biogeochemist perspective?
I would love to.
Thank
I
you.
think a lot about oysters and nitrogen cycling, and I love Boze's last little tidbit there, well, before Ed's question, about the goal of restoring those oysters by 2025, because in Chesapeake Bay, that is when their TMDL is expected to be met for nitrogen. So just reminding everybody of the importance of oysters for nitrogen removal. Let me share my screen here. And then, okay. So thanks everyone for inviting me today. I'm thrilled to share my work on oysters and nitrogen removal with everybody. I was among the first people to measure nitrogen cycling in oyster reef ecosystems, and that was about 13 years ago. Since then, I've been able to do similar work in North Carolina, Virginia, Maryland, and Georgia, and hopefully soon in Florida. The concept of oysters and other bivalves being used for nutrient management has gained a lot of attention since about 2005, when one of the first papers came out from Sweden using mussels to treat wastewater. Using mussels to treat wastewater. Since then, we're seeing more and more projects pop up, more interest in this, including in Rhode Island, New Jersey, North Carolina, Maryland. So today I'm going to spend some time sharing my work and insights that I've learned from those other measurements that I've done to illustrate the importance of oysters for removing nutrients and how they actually do that. I will also share some economic evaluation of the nitrogen removal services that can be provided by oyster reefs. And while my focus is on reef restoration, I will also talk a little bit about aquaculture. So while oysters are the star of our show, before we can talk about oysters, we need to talk about nutrient pollution. Eutrophication is a global problem. This map is a little old now, it's from 2008, but it shows the human footprint. So that's the different colors, so how much anthropogenic activity is there. And then the dots represent coastal systems that have symptoms of eutrophication. Eutrophication resulting from nutrient pollution. It's characterized by algal blooms, which lead to low oxygen events, fish kills, increases in turbidity, and then even reduction of benthic habitats. In the US, 65% of our estuaries are classified as moderately to highly impacted by excessive nutrients. Here are a few photos of what eutrophication looks like. We have opportunistic algal macroalgae, fish kills, hypoxia, and then in Florida, we've begun to say that eutrophication is what's turning our water guacamole colored green. It's very different than the pea soup green I'm used to thinking about, but. And one of the main, when we think about eutrophication and that change in water color, we have to think about nitrogen. So nitrogen is the main pollutant of concern for eutrophication in coastal systems. Too much of it is really considered a bad thing. If you're like me, it's a little bit tricky to think about nitrogen as a pollutant because it is an essential element for life. So I think about nitrogen for our coastal systems like I think about coffee for myself. I need a little coffee to start my day, but if I have too much coffee, I'm going to talk like this and my system's going to crash and no one's going to hear war rooms. That's how nitrogen really is for coastal systems. They need nitrogen. It supports primary production at the base of our food web. It's fundamental to our cells. So with a little bit of nitrogen, we can have that healthy ecosystem that we're used to seeing, that we want. Too much nitrogen is when our systems become eutrophic, and they turn that guacamole colored green, with all these other consequences, with fish kills, loss of benthic habitat, poor light availability. So it's really, these are the consequences then of that too much nitrogen. Managing nitrogen has then become a main goal of a lot of coastal watersheds. We have engineering solutions to do that, which are through wastewater treatment plants and stormwater ponds. These are ways to capture and process and clean that water before it would enter the coastal system. However, these systems are inefficient at non-point sources of pollution. They're really good at treating point sources when you can say, like, funnel all of our runoff to this system and treat it. But in a lot of agricultural watersheds like Chesapeake Bay, like down here in Florida, they're non-point sources of pollution, and they're more difficult to capture. But lucky for us, there is a natural process that can occur in sediments that can help remove nitrogen from water, including that nitrogen that comes from non-point sources. That process is denitrification. Denitrification is a natural nitrogen sink. It's the microbially mediated reduction of bioavailable nitrogen. So that nitrogen that's nitrate in green on here, it's green because it feeds the algae. And then it converts it into N2 gas. N2 gas is basically inert. So you can think of denitrification like respiration, but using nitrate rather than oxygen. So this process is a way that we can transform the nitrogen that would once feed the algae and turn it into N2 gas, essentially removing it. There are three pieces of the environment that can control the rate of denitrification. For denitrification to happen, you need to have carbon, because that's the energy source for the microbes. You also need nitrate, because that's what's being reduced to N2 gas. And then denitrification is performed by facultative anaerobic bacteria, so they need low oxygen conditions. If there's there is plenty of oxygen, they won't use nitrate, they'll use oxygen for respiration instead. When looking at the historical record from Chesapeake Bay, Grace Brush suggested that the one solution to really reduce our nitrogen pollution problems there was to restore habitats that have high rates of denitrification. These habitats are salt marshes, seagrass beds, oyster reefs, and they have been lost to development, pollution, and deterioration of water quality, and overfishing in the last century. So my work is then focused on identifying which habitats are hotspots for nitrogen removal. So the majority of my dissertation work focused just on that. And here are the big results. That we do see that there is, in fact, increased denitrification, which is on the y-axis here, with those habitats like salt marshes, oyster reefs, seagrass beds. And they increase increased denitrification compared to our intertidal flats and subtidal flats. So having these habitats in the system will then provide many benefits in terms of denitrification. The one thing that is different about some of these habitats, and like what Boze just talked about with oyster reefs, is that they have this three-dimensional reef structure, and they're also made up of filter feeders. So they can provide different ecosystem services and different benefits than having salt marshes or seagrass beds. So because of all this growing interest in oysters and their ability to filter water and to remove nitrogen, we are seeing more and more people asking questions about using oysters as the solution to our pollution problems. These are just a few headlines that I pulled from a variety of different reports. How mussel farming could help to clean fouled waters. Stanford research shows value of clams and mussels at cleaning dirty water. And then could oysters be used to clean up Chesapeake Bay? There's even this cartoon from the Washington Post showing that nitrogen filtering oysters are the sunken treasure to cleaning up Chesapeake Bay. And because of this promise of oysters, we're even seeing programs grow throughout coastal systems, like the 10 billion oysters for the Bay project in Chesapeake Bay, and then the Billion Oyster Project in Long Island Sound. Everything has to be bigger in Chesapeake Bay. They just needed 10 billion. But before we can talk about the way that oysters really interact with nitrogen pollution, we have to talk about the nitrogen cycle. I usually don't put a nitrogen cycle figure up in my talks because people see these and they kind of takes them back to college and thinking about all the different processing and transformations of nitrogen, but that's what gets me really excited. Nitrogen is mostly in our atmosphere. 78% of our atmosphere is N2 gas. That's where we want nitrogen. Through a variety of processes, that nitrogen can be transformed then back into N2 gas. But on this figure here, you see that it's not really a cycle at all. And the nitrogen cycle is actually more of a complex web with all these different reactions mediated by different groups of microbes. And they're all affected by the abiotic and biotic conditions in the environment. When we think about the global nitrogen cycle, it's important to remember that human activities have at least doubled the amount of nitrogen, fixed nitrogen, so in this organic form, that's cycling through our ecosystems. And that's because we've been able to synthetically create fertilizer. So how is it that oysters then interact with the nitrogen cycle? Here we have our oyster reef located on the sediments, and the top surface of the sediment is aerobic, so there's oxygen there. The bottom sediment is anaerobic, or anoxic. We have the nitrogen that comes into the water, and it feeds the algae, and it grows. We get these blooms. The oysters graze that algae, repackage it as their biodeposits on the sediment surface, and they also take up some of the nitrogen that was in the algal biomass into their shells and tissues, and they use that for growth. The portion of the nitrogen that is now in their biodeposits on the sediments is juicy food for microbes. The microbes can process that, mineralize it, and create ammonium. Ammonium is a bioavailable form of nitrogen, and that ammonium can flux out of the sediments where it could then be used to feed algae again. or it can stay in the sediments. When conditions are right, it can be nitrified to nitrate. Nitrate is another bioavailable form of nitrogen, and that can flux out of the sediments and again supporting more algae growth. But if we focus just on the sediments, then that nitrate could end when there is more biodeposits available, so more carbon. That nitrate can diffuse into the low oxygen zones in the sediments where denitrification can occur. And that denitrification, that N2 gas production, is then that removal of nitrogen from the system. So really, it's this benthic pelagic coupling of oysters that can move nitrogen that was once in algal biomass to the sediments, where it can then be processed by microbes to convert it into N2 gas. There is some more recent studies that are looking at the oyster microbiome, so the denitrifiers that are associated with the guts, the gills, and the shells of oysters, and they play an important role too in denitrification by creating these little anoxic microzones. So can oyster reef restoration remove nitrogen? The expectation is yes. We see that oyster reefs have high rates of denitrification, but there are factors like salinity, temperature, food, the amount of algae that's there for them to graze on, location, predators can all affect their ability to remove nitrogen. Some of my work from North Carolina really tested this to see how restored oyster reefs are, if restored oyster reefs are able to remove nitrogen through enhanced denitrification. We were lucky enough to work on oyster reefs that had been restored by John Grabowski and colleagues in 2005. When I did this work, those reefs were about 10 years old. These reefs were located in a protected area and in different habitat contexts. So we had oyster reefs that were restored next to salt marshes, oyster reefs that were restored next to seagrass beds, and then oyster reefs that were restored isolated on a mud flat like you see here. So this gives us a chance to kind of capture all the variability about where you might want to restore an oyster reef. Here are our results. Again, this is our denitrification rate. So we have our sediment, that's our control, just bare sediment, no oysters. And then we have our restored oyster reef. And we see that the oyster reef does increase denitrification relative to the control. So we see this enhancement in denitrification when the oyster reef is present. The one thing that I don't have on this is that importance of that location. What we saw was that oyster reefs restored on mudflats had higher rates of denitrification or enhanced denitrification than oyster reefs restored on adjacent to salt marshes or seagrass beds. So I think that what happens is when you restore an oyster reef next to a salt marsh or seagrass bed, it becomes functionally redundant in terms of their nitrogen removal ability. So that's something to, so the context that you restore reefs is something to consider when you're evaluating then the services that you expect to get in terms of nitrogen removal from that habitat. So lucky for us that there is a way to determine an economic value of the nitrogen removal service that oysters provide. That value can be from cost of replacement, so having to remove that nitrogen through other means like wastewater treatment plants, or through creating agricultural or urban BMPs. If you're lucky enough to live in a state that allows for nutrient trading credits, you can also trade nutrients. So what I have here is the dollar values for these different ways to evaluate the nitrogen removal services. The wastewater treatment values that I have actually came from Florida. They come from a variety of different counties located, coastal counties in Florida, from a study by Baker and colleagues in 2015. It was focused on clams, but the habitat, I mean, the dollar value is the same for the wastewater treatment plant. The agricultural BMPs and urban BMPs are from Long Island Sound, and then the nutrient trading credit value is from North Carolina, where I know they do trade nutrients. For our data, we have to, and for most of these assessments, you have to look at the enhancement. So how much denitrification has increased with the oyster present, because that bare sediment does provide some denitrification service. So in our study, we found a 23 kilograms of nitrogen per acre annual increase in nitrification because of the oysters. This is about the same amount of nitrogen that is in one 50-pound bag of fertilizer. So we can use this enhancement in denitrification to then calculate removal services from the, and here we can see how much dollar value per acre per year that those oyster reefs provide. So this value can range from $176 to almost $8,000 per acre per year. And this is important when you're thinking about the return on the investment of your oyster reef restoration project. So are oysters the solution to our pollution problems? The majority of the literature says yes, oysters are hotspots for nitrogen removal. If we put this into the nitrogen removal services that I just talked about into context of Florida, and we'll use the St. Lucie estuary as an example, they have a BMAP to meet a nitrogen reduction goal of 143 megatons of nitrogen per year. To achieve this goal through oyster reef restoration alone, it would be required to restore 6,190 acres of oyster reefs. That's about 12 football fields worth of oysters. So restoring oysters is expensive. We might not even have the space for all of those oysters that we would need to meet the nitrogen reduction goals for the St. Lucie estuary. But there is a way that we can get the benefits from oysters in terms of filtration and denitrification in an economic framework, and that would be through aquaculture. As aquaculture is growing, I think it's important that we think about getting the environmental benefits of bivalve aquaculture. They should provide the same water quality filtration services as a restored reef. Also provide an additional nitrogen removal service through that bioextraction component at harvest that a lot of our reefs don't provide because we set them in sanctuaries, because we want all those other ecosystem services that Boze just talked about. And aquaculture also contributes to the local economy. It also helps to meet the current demand for oysters, because let's face it, everyone wants to eat their oysters at their fancy oyster restaurants. When I started to look at what we knew about oyster aquaculture and the nitrogen cycle, studies were inconclusive. There were some studies from floating aquaculture that show that oysters do not significantly increase rates of denitrification. And then some rack and bag, some more on-bottom oyster aquaculture farms that showed enhanced but limited denitrification at oyster farms. So I set out, so we're hearing all this, that oyster aquaculture is good, that we can get all these benefits, but this is really what we knew about it in terms of denitrification. And I set out then to look at a larger scale oyster aquaculture farm. This aquaculture farms that I looked at all produce more than 1 million oysters per year. And they all used this off-bottom cage aquaculture that you're seeing here. These were all located in Virginia and covered a variety of different salinity regimes. What I found was that salinity did not matter. In either instance, oyster aquaculture increased sediment denitrification relative to bare reference controls. So I pooled that data here. You again see our denitrification rate on the y-axis and our different oyster or aquaculture or no oysters on the x. And we see that there was a 225% increase in oyster denitrification with aquaculture compared to no aquaculture. Our reference sediment for this study was part of the aquaculture farm, but it had never had aquaculture on it. When I put the denitrification measurements in context of how much nitrogen was removed at harvest at these larger farms, the total nitrogen removed, 64% of that came through harvest. So there was much more nitrogen being removed through harvest than through enhanced denitrification. In fact, the amount of nitrogen removed through harvest is now traded in Maryland. Just this past fall in 2019, they issued the first nutrient trading credits for aquaculture through that bioextraction. So that amount of nitrogen that's harvested in the shell and tissue. This recommendation came from an oyster BMP expert panel that was charged with establishing the nutrient reduction effectiveness and deciding on the framework to using oysters as a BMP. based on the best available science. I think it's really exciting that we've been able to trade oysters for nutrient trading credits. And I think that there's a lot of potential here for continued growth of this concept for water quality benefits. In fact, later today, I'm hopping off this call and having a call to focus on actually how that deal was brokered, because this is all I know about it from Facebook. But what about denitrification? Can we get credit for that too? Well, no, not yet. There's still a lot of variability in denitrification measurements, and that results partially from different incubation methods, where we have like batch style incubations with using isotopes, whole core incubations, slurry incubations, and then even larger tray incubations with whole pieces of oyster reefs. And then the methods to measure denitrification vary, where there's an N2 to argon method, which is more of the like traditional or actual denitrification rate. Then we have potential rates where you add nitrate, and you can measure those, and you have isotopes too, using N15 nitrate as tracers. The general consensus, though, is that the N2 to argon method is the best way to measure actual denitrification rates with oysters. What does all this mean for Florida? So I've looked through the literature, and from what I can tell, there are only 3 sets of data that relate to oyster denitrification. One is one that I've collected since I've been down here. One is from my PhD supervisor, and then another study was done in Mosquito Lagoon by Lisa Chambers at UCF, and she used these potential slurry incubations. So even those data would not fit with our criteria for establishing a way to trade denitrification. So I look forward to getting the chance to populate this chart, because there's a lot of places where I know all you guys are thinking about restoring oysters or doing oyster reef restoration. And in some of these estuaries that have TMDLs and BMP and BMAPs. So figuring out when, how, and if we can use oysters to help meet our nitrogen goals is really part of what my work is focused on. So overall, I think that oysters are really part of our solution to our water quality problems. And they can be useful in developing tools to help reduce nutrients, but they should not replace existing management strategies. That's why they are part of the solution. With that, I'll take any questions, and thank you for your attention.
Hi, Ashley, that was a great presentation. I'm curious that some of the bioextraction, they give credits for shell removed. Wouldn't the shell eventually make its way back into the bay system?
Yeah, I think the best way to do it would be if me down in Florida are eating oysters from Virginia. I'm not sure entirely how they are. So it depends where the shells end up, and even the meat, like who's doing the where. So I'm curious to hear how they, more on that call today, about how they brokered this deal.
Interesting, thank you.
Hey, Ashley, it's Ann. Thank you for this. I had a question further back in your presentation. You said something, maybe I didn't catch all of it, is that functionally extinct nitrogen removal when oyster reefs are adjacent to salt marsh and seagrass. Can you explain that again, please? What?
Functionally redundant. So we didn't see an increase in denitrification when we had oyster reefs with seagrass beds. So the seagrass beds alone provide the same denitrification enhancement that the oyster reef did. So it was sort of like, yeah.
Okay, thank you. Yeah. Mm-hmm. Are there areas in Florida that you're looking to do additional research?
I don't know, Ann, what do you have in mind?
I don't know, maybe we can talk. I think it's, helpful that as, restoration is being planned, restoration projects, if there's a way to design projects that can help meet research objectives, we should try to do that and work cooperatively with the researchers when we're designing projects.
Yeah.
rather than having it be an afterthought. Oh, now can we do this research? So
yeah, and I think that that's sort of the best way to get at that enhancement for denitrification. If you know what the baseline was, and then you add your oysters, then how does that change? So yeah, I'm all ears. Happy to talk locations.
Hi, Ashley. This is Steve Durham, FDEP. My question is, do you have a comparison? You noted the increase in nitrogen removal comparing aquaculture with bare sediment, and then you had a separate study comparing natural reefs with a variety of other habitats. Do you have a comparison of the relationship between natural oyster reef with the aquaculture removal benefits?
So my master's student is currently doing that. It's also from Virginia, but she's working on those data right now. So stay tuned.
All right. Yeah, because a couple other things I was wondering is, do you have any idea what the effect of, like, an oyster reef obviously has more three-dimensional relief, and there's a lot of sediment packed in there that could potentially harbor these, microbes that you're talking about. And then the shell itself, I've noticed a fair amount in various samplings that I've done that there's lots of pore water flowing through oyster reefs, like through the matrix of the reef itself. That might be something that's different between a bare sediment and a reef structure. Is there, because some of those benefits, if there are differences there, might not be associated with the living oysters themselves. Perhaps it's associated with the structure of the reef.
Yeah, so two things to this. So we did a study, I can send it to you. It's in MEPS, where we looked at this exact question, where we have a bare sediment control, and then we had like oyster reefs. So they had all the structure, but none of the function. They weren't filtering at all. Mm-hmm. And then we had our live oysters. And we saw that both of those treatments increased denitrification relative to the control, the bare sediment. And overall, 40%, if you think of a whole reef with like the functioning of the filtration and the live oyster and just that shell structure, 40% was just from the structure, and the other 60% was from the filtration function of the oysters.
Okay, interesting.
I think too that as we're starting to really dive deeper into this and like geek out a little bit about all the microzones and cool places that denitrifiers could live on these oyster reefs, having like larger trays with pieces of reefs in them and getting at sort of both the diffusive and advective fluxes is a cool direction to go.
All right, thanks. And if I could ask, sorry, I don't mean to dominate, but one more thing that I was wondering, you were talking about this obviously in relation to eutrophication, and I know that there's a relationship between clearance rates and oyster growth and that kind of thing associated with food availability, which is essentially, you see an increase in food availability due to eutrophication initially, but then there seems to be a point at which food availability, like the oysters are kind of overwhelmed or something, and oyster growth no longer benefits from additional, food levels. Is there, in a lot of our estuaries that really might need nutrient reduction the most, perhaps the oyster, feeding rates are going to be a little lower than if the food availability was a little more moderate.
Yeah, I think that's a really good point, that where we restore these reefs, does that match up where we need that service, mm-hmm. right? Like in those estuaries where we have a lot of really dense algal blooms, we could spend money to restore reefs there, but we might not actually get that benefit. because they might be saturated and, this bottom is still anoxic. So I mean, I think it's a good point and something to consider. I don't have a good sense on I don't have data for any of that, but it's definitely something I think about, is restoring where and for what water quality services are we after.
All right, thank you.
Could I perhaps add a comment?
Yeah.
Boze here. Ashley, I appreciate your comments about the different methods.
I did that just for you. No.
Yeah, good for you. So yeah, that's the kind of reason I pointed at the Oyster calculator at what I consider to be the empty column on the right of the filtration and fish production. We don't yet have denitrification, and one of the reasons the reason is that we're still seeing a lot of variation between sites. So Ashley, you described a study where you were looking at aquaculture and oyster reef, and about 40% of the denitrification from oyster reef, 60% in terms of rate associated with aquaculture. We've done a study 2015 in Rhode Island where it was exactly the opposite. It was 60-40 for reef and aquaculture, but both pretty high, which is encouraging. But it seems that there are factors affecting the rates of denitrification which we don't quite understand. And one of the ones that both Ashley and I are involved in trying to figure out is how much of that is due to technique. Because if a reasonable amount of that variation is just a matter of how it's being measured, that's easy to fix. We can understand that. There's bound to be other factors that are environmental that are influencing, denitrification rates, and I'm not sure that we have a handle on all of those yet. The work Ashley's doing is fantastic at trying to nail some of those down. But I'm kind of mildly optimistic that the technique issue has at least a portion of that variability associated with it, and we can eliminate that. So I'm hopeful that we're getting closer. Mm-hmm.
Ashley, this is Anne again. Are there thresholds or characteristics of a reef as far as size, structure, percent live dead that have to be calculated into the nitrogen reduction, DNF?
Yeah, so I haven't really, I've looked a little bit at oyster density, a little bit at the age of the reefs that go into there's the density matters, suggestive of a threshold where you get too many oysters and then you won't get these, you sort of turn on other processing. So DNRA, which is the opposite of denitrification, where you actually regenerate ammonium instead of denitrifying it to N2 gas, can happen at really, really high densities. So but I think it's going to be system specific too for what that threshold is.
Yeah, just wondering how folks are doing the nutrient trading then, or looking at that and managing a reef to make sure that the denitrification is happening at a rate that is needed.
I think there's, tons of challenges with trading denitrification, which is why they haven't yet. But
and that's one of the benefits of making this something that can be modeled. So at the moment, any trading is specific to areas that have measurements at a density that's sufficient for people to be confident that they know what the denitrification is. And we can't yet apply those denitrification rates to another system because we don't understand what's driving the variability well enough. So yes, trading at this point means you need measurements from that system.
Yeah, so for the Chesapeake Bay, their trading of the bioextraction from the aquaculture, I think they used 8,000 points on nitrogen and phosphorus content in shell and tissue to be able to make that regression of size to nitrogen content. So
This is Steve Durham again. So this may be a naive question, but having, like, I know, you know, there are optimum ratios of like carbon, nitrogen, phosphorus for different biological processes. And if most of the fertilizer that's being produced is nitrogen, could some of the variation be related to, you know, there's some other limiting pollutant or nutrient that is changing the amount of nitrogen that can be processed?
So yes. So if you're in a system that has like low nitrate in the water column to begin with, you might not have as much denitrification as you would in a system with high nitrate in the water column, because it's not available then. They have to process it to get there. So that would affect the rates. There is sort of this like, so the denitrifiers are microbes, so they're going to hit a limit at some point where they can only denitrify so much. So I think that once we start to constrain all that, we'll have a better sense of these real driving factors, some of which are like ambient nitrate concentration.
All right, thanks.
Thanks so much, Ashley.
Thank you.
Great questions as well. It looks like Anne has shared the BMP nutrient panel report that you mentioned in the box. Everybody's aware of that. And Ashley, would you be willing to share your presentation slides as well? Excellent. Sure. Thanks so much. Okay, so then Chris Shepard with the Nature Conservancy is up next, presenting on Gulf of Mexico Ecosystem Service logic models and socioeconomic indicators, or the GEMS program. And Chris, for your information, several members of this group have heard a little bit about, I think, the GEMS in the past. Correct me if I'm wrong, anybody, but we're looking forward to hearing more and learning about how it impacts ecosystem services.
All right, can you hear me okay?
Yes.
Okay, I had my video on earlier, but it was getting a little bit glitchy, so I'm just gonna use my voice and my slides for now. Thank you for the context that some folks have seen a GEMS presentation. I kind of figured that, and so I tailored this a little bit to be more about kind of connecting you all with resources that might be helpful for your group. So let me make this big. All right. So for those of you who have not heard of the GEMS project, GEMS stands for Gulf of Mexico Ecosystem Service Logic Models and Socioeconomic Indicators. So good thing we came up with that nice little project title of GEMS. That's a mouthful. But the project is an NAS Gulf Research Program funded project. And the first entire year of the project, where we figured out the process that we'd like to apply, focused on oyster reef restoration. We chose oyster reef restoration because it's so prevalent with the number of projects being taken place and funded in the Gulf of Mexico. And also because a lot of the science on the ecological outcomes and kind of the more restoration focused outcomes it is in existence. And also there's already a lot of monitoring and metric protocols for more of the restoration outcomes of oyster restoration projects. What GEMS really focuses on is the social and economic outcomes of oyster reef restoration. So a lot of the logic models that exist out there, I've been on other projects where we've done oyster reef restoration logic models with some of the folks on this call today even. They kind of, a lot of them tend to end at the restoration outcomes. And so GEMS really focuses on carrying that all the way through to think about human benefits and well-being. So the GEMS project is a collaboration between Duke University, Hart Research Institute in Texas, and the Nature Conservancy. So I think there's a slide of the team. So you can see kind of the folks that comprise the GEMS team. there's a whole host of folks working at Duke and at HRI. And then on the TNC side of things, I'm the lead PI from TNC, and connect this work through our 5 Gulf State TNC chapters as well to kind of connect to the oyster work that's taking place within TNC. So, you know, the need for a GEMS type project kind of exists at multiple levels, and in terms of trying to understand what the measurable outcomes are of oyster reef restoration projects and all other types of restoration projects, so that we can kind of roll up projects and have a better idea of how they're actually contributing to human well-being. And there's not a ton of guidance out there as far as how to do that, what the standardized metrics would look like. And it sounds like some of the GEMS products that I'm going to share today can kind of directly inform some of where your Florida oyster recovery efforts are at as well. But the need for this type of work is kind of pretty well documented. Some of the publications that kind of note the need for this type of work are shown here. But really across all of our organizations, we really have a need to better quantify the human well-being outcomes associated with our restoration work, both in terms of if you're a funder wanting to fund work and understand what you're getting for your return on investment, and also if you're just planning a restoration program, kind of be able to roll up the outcomes to communicate them to partners and to the public. So the Gulf of Mexico, because of the amount of funding that is available and being the amount of restoration projects that are being implemented, there's a real need for this kind of information even right now. And so, you know, the goals of the Restore Council very much so focus on the community benefits and benefits for people. And so that's why we developed the GEMS project to kind of really take a closer look at, well, what are the kinds of outcomes that are likely to be seen from restoration investments, and are there any that are measurable and closely enough tied to the restoration project where they could be required for monitoring. And so the GEMS project was developed to start to think through kind of what the process would be to figure out what those outcomes are and how we would measure them. So the GEMS basic approach starts with developing the logic models. And so at the very beginning of the project team did sort of like a draft logic model, and I'll show one of those in just a minute here. And then we had a series of workshops in each of the 5 Gulf states to kind of talk through those logic models. Some of the folks that are on this call today participated in the Florida workshop. And so once we started to kind of map out the logic models and carry that all the way through to social and economic outcomes, then we started to identify, well, what metrics would we use to assess those outcomes and to measure them. So let's see here. The ultimate goal, as I mentioned earlier, is really to allow people who are funding restoration, people who are planning projects and programs to restore whole systems or across multiple systems, to be able to compare what they're likely going to get out of these projects across a variety of different types of projects. This approach also allows us to understand where there is strong scientific evidence for social and economic outcomes versus non-existent or weak leakages, and I'll show you a little bit about that. And then also just tracking performance towards social and economic goals. A lot of projects don't have those types of goals as part of the project or part of a restoration program. But we think some of the metrics that we've identified through the GEMS process can kind of help inform how to set those goals. So as I mentioned, The first year of GEMS focused entirely on oyster reef restoration as we developed the process for this project. So we had a series of workshops in one bay system in each state, and we chose a diversity of estuaries so that we had a mix of kind of socioeconomic circumstances, levels of oyster harvest, levels of oyster health and condition. So we wanted to have a variation across the bay systems. But in each of these 5 estuaries, we held a workshop and brought folks together to talk through the draft logic models, identify the outcomes that were important within each of those estuaries, and again, focusing on the social and economic outcomes. And then starting to work through metrics that and identify those metrics that are feasible to measure the outcomes both at the project and at the regional scale. To be able to kind of organize the process and the conversation around oyster reef restoration, we divvied up different kinds of projects into these 6 classes. They're not perfect classes, but they're classes nonetheless. And we have information on the GEMS website about kind of how we divided these up, and we actually developed logic models for each of these different types of restoration approaches. So this is the general oyster reef restoration ecosystem service logic model. Many of you are familiar with these, work with these all the time. So there's really not a whole lot that's new here for folks that have worked on these types of models to try to understand how interventions in the system can impact the system. But what we did here was we kind of divided them into the more ecological outcomes. So the green outcomes here, those are typically, you know, the outcomes that are the restoration outcomes. So a lot of logic models tend to end there with some consideration of some of the more blue human activity outcomes and kind of the relationships between the restoration outcomes and people. So for the GEMS project, for each of the restoration techniques that we've developed these models for, we're really concerned with getting over here to the right side of things in the yellow boxes for the socioeconomic outcomes, so those that are impacting human well-being. And for each of these logic models, there's a whole evidence library that's been developed to inform the development of this model, and I'll show a slide of that in just a minute. So what I wanted to do today, because some of you have already heard about GEMS, was kind of just direct you to some of the resources that might be able to help you skip a few steps when you're thinking about metrics and standardizing monitoring and metrics through the work that you're doing right now in Florida. For a lot of the green boxes that I showed on the model, the ecological outcomes, there's a lot of standardized guidance that exists, and I kind of poked around a little bit on some of the resources that were shared for this call and saw that you have those kind of at your hands already. Several folks on here probably even contributed to the development of those resources. So as far as metrics for social and economic outcomes, all of our findings and collaboratively developed metrics lists that we developed through GEMS are available on the GEMS website. And I can share this PowerPoint after the call today so that you can access the links. It's really easy to find though. If you just Google Duke and GEMS, it kind of is the first result to pop up. But what's available on that website that might be useful for planning kind of at the bay scale and then across the state potentially is each of the logic models that we developed are available, and they're available in an editable version. So depending on which system you're working in and which estuary, you can kind of delete outcomes that are not relevant or for a specific project, you can kind of edit that logic model and use it in proposal development or developing a restoration program. There's a couple different ways you can use it, and there's guidance about how you can use it on the GEMS website as well. I mentioned the evidence library, and that was developed by the Duke team. So they did a literature review to basically validate each of the steps along the chain in the logic models. And so what that did was help to highlight what parts of the logic models, are very strong, and what parts of the model are more weak, as far as is there an evidence base for linking the restoration project to the outcome, or does that evidence not exist? And so that's been characterized every step of the way through this logic model. So you can kind of see here each of the numbers that's on the lines connecting the boxes. That is actually, I'm going to kind of go out of here. This is the Duke GEMS website. But this is the evidence library for oyster reef restoration. So it contains the logic model, and then each step along the way has a number, and you can scroll to that section of the model for each link, and it kind of gives a summary of the evidence for that. And so it summarizes how much evidence there is and how strong of the connection is between the restoration intervention and that part of the logic model leading to the outcomes. So throughout the process, when we were developing the list of metrics, which I'm going to share with you next, that we came up with for the vetted list for oyster reef restoration, there's so many, there's a lot of metrics information already out there. So some of that I mentioned earlier for more of the ecological outcomes, but this is just a list of some of the resources that we reviewed when we developed the metrics list. And really the benefit of the GEMS project was that we vetted the metrics with scientists, agency folks, and practitioners. So we wanted to make sure, well, yes, if there's a strong connection between oyster restoration and some sort of socioeconomic outcome, when we're identifying those metrics, we wanted to make sure that the metrics were feasible for kind of folks who do this type of work on the ground to be able to apply the metrics in their work. We also considered the SMARTS criteria. That's to kind of think about, well, are they relevant? Are they time bound? Are they specific? When we were thinking about which metrics to include, so we included this type of criteria assessment in each of our workshops to consider the metrics. So ultimately, we came up with 2 tiers of metrics. So tier 1 metrics were those that the folks that we collaborated with in the workshop series and other oyster restoration practitioners really felt like they were feasible to require them or strongly suggest that they be monitored for projects. So tier 1 is kind of those that we feel like it's a reasonable request to have them be monitored and reported on for oyster restoration projects. And tier 2 is really those that are, would be great to have, but are a little more difficult as far as time and capacity and money to be able to measure them. So we have tier 1 and tier 2 metrics, and we have those for both the project scale and the program scale. So I'm going to share a couple of those with you. And each of those metrics are informing on these boxes on the right here. So economic activity, public infrastructure protection, cultural values, and human health. So the first one, the first kind of list of metrics are all related to economic activity. And so at the project scale, you know, it's pretty simple, but it's actually something that is not always easy to find. It's just the number of jobs and created by the projects themselves, and as far as designing, implementing, and constructing them. And so Boze had a slide earlier that kind of showed some of the jobs information. So some projects, some funding sources require this to be reported and some don't. And so what we're really suggesting is just kind of like a standard reporting for jobs moving forward for oyster restoration projects everywhere, so that we would have, you know, a more comprehensive data set on that. Boze also showed the slide from the Half Moon Reef study, which we led in Texas a few years ago, which looked at recreational fishing benefits due to that project. And so for Tier 2, this came up quite a bit across all of the workshops. Recreational fishing expenditures and jobs. That's something that not all practitioners have access to funding to do that type of study. We were able to fund the Half Moon Reef study with some private money, but it wasn't that expensive. It was about $15,000. We contracted with Texas Sea Grant, and we were able to get some really useful information out of that project that's been shared across the U.S. and in other countries as far as the recreational fishing benefits associated with that Oyster Reef project. So for the project scale, they're really metrics that the project implementers can assess these metrics, measure them for themselves. For the more of the program scale for economic activity, it's really an economic impact analysis that we are proposing, and it's hard to connect across projects and assess this information. But what the goal is there is to measure the change in economic activity, so change in jobs, income, taxes, for example, given the implementation of a suite of projects. And so for this particular, the program scale metrics, we're working on some protocols for that right now, so that we can actually provide guidance on how to do that. We're actually working on protocols for all of these metrics. That wasn't originally within the scope of what we were proposing to do with GEMS, but it became pretty clear when we started sharing this work around that folks said, okay, this is great, but how do you do it? So we are developing protocols for all of the metrics that I'm sharing in this matrix. The next one's public infrastructure protection. So assessing how many miles of shoreline are being protected by an oyster restoration project. That one's a little bit more straightforward than some of the economic activity work, but I'll share one of the protocols for that in just a minute. For cultural value, there's a couple of metrics associated with cultural value. This is another one where there's a big need for the protocols. So how would you assess cultural value? So many of us are ecologists by training. You know, we don't have a lot of experience in these kinds of metrics. And so the Duke team right now is developing the protocols based on the literature and based on some survey work that's taken place for other types of restoration projects to come up with actual questions that folks could use for monitoring for this metric. And that's all going to be on the GEMS website as well. One issue that came up quite a bit throughout the year focused on oysters through the GEMS project was the connection to human health, because that's something that everybody knows is there, but we don't really know how to link it just yet. The science is not to the point where we can link a restoration project to a human health outcome at the project scale. So in the evidence library that we developed for this project, and then also in our discussions with all of the working groups about the various metrics, When we got to the point of, well, what is the strength of the scientific evidence for this linkage, we kind of couldn't include any of the human health outcomes in our recommended list of metrics. So the good thing that's come from this project is that we've kind of highlighted these 3 key areas of health, community resilience, and equity as key gaps where we need to have a lot more science to be able to link between restoration and these outcomes, because we know it's important as far as, you know, oysters providing a source of protein communities or contributing to fish production, which is a source of protein for communities, as far as water quality impacts and linking that to human health. So there are linkages there, and we know they exist, but we weren't able to, they just aren't quantifiable yet enough to be able to link them and include suggested metrics in our metrics list right now. So I mentioned briefly that we're developing protocols for each of those metrics that I just shared with you. And so each one has kind of like a preferred approach and a secondary approach. We're trying to organize these based on kind of budget considerations, similar to the way that other monitoring guidance exists for some of the more ecological outcomes of oyster reef restoration. So all of this information exists on the Duke GEMS website. This is an example for the project level protocol for cultural values. So just looking through the literature, we found some of the kinds of questions and approaches that can be used to try and understand cultural value. It was one that came up as important across all of the workshops that we held in all 5 Gulf states, because it really contributes to sense of place and sense of community for places that have oysters or have historically had oysters within their bays and estuaries. And so just developing the methods to be able to track that over time is a bit of a new area for a lot of folks involved in oyster restoration. So all of that work that we did for oyster restoration was phase 1 of the GEMS project, and now we're in phase 2 right now. And so we decided to look at as many other restoration approaches as we could for the phase 2. So other types of habitat restoration, hydrologic connectivity, recreation enhancement, and water quality. So we have logic models developed for all of these, and we're going through a slightly different process because we're doing multiple restoration approaches this year and not just oysters. And also with everything being virtual due to COVID, it's a very different phase. But we will ultimately end up with the same products for each of these restoration approaches as we've developed for the oyster work. We are nearly finished with our practitioner's guide to how to use the GEMS metrics for oyster reef restoration. So that'll kind of walk you through a lot of what I shared today, along with some specific examples, and then hyperlinks within this document to all of the protocols. So it'll be a pretty good primer on how to think about measuring for social and economic outcomes. Where we are in the process right now is that we're finishing the models and the metrics for each of those phase 2 restoration approaches, finishing up the practitioner's guide. We're also developing a companion guide for funders, thinking about how to fund work, and kind of requirements and suggested monitoring protocols. And let's see here, for the next project, we're kind of thinking about how GEMS can continue on after this project ends, which will be later this calendar year. And what we'd like to do, specifically TNC and Hart Research Institute, is to find some projects to apply the GEMS metrics to. So kind of like feasibility testing of applying those metrics to projects, and also some programs. So looking across like a regional restoration program or a state level oyster restoration program to begin to test how you would apply both the project and the program scale metrics to see if adjustments need to be made and if what we prescribed as feasible truly is feasible. So with that, I'll leave you with the website for the GEMS project. Feel free to check that out. You can download the evidence library. You can download the logic models and really, you know, kind of tailor those and use them for proposals, for project reporting, and yeah, and my email is here as well in case you have any questions. But I'd like to thank you all for the opportunity to share with your group today about the GEMS project, and I'll open it up for questions.
Thank you, Chris. This is Katie. Just curious, you mentioned that a lot of projects don't currently have social or economic goals, and how these GEMS products will be helpful for setting those and tracking them. Just curious, are you seeing more funders requesting those types of metrics and proposals for oyster reef restoration?
I think it's definitely starting to move that way, depending on, you know, the funder. And we've had quite a few conversations with folks, restore staff in particular, about kind of the feasibility of the metrics that we identified and how those could be included in further rounds of funding. So I think, you know, it's not attributable to the GEMS project because that's kind of taking place now, but definitely over the past, you know, five years or so, I've seen it moving in that direction, which is a good direction. But just the more standardized we can be, the better. And the goal is to kind of have these metrics be standardized in kind of the way that other oyster restoration metrics are standardized. And so when folks are, you know, wanting to stand up a project, or someone's kind of new into restoration, they have access to so many resources now that has kind of standardized the approaches, and we just want some of these metrics to kind of be on that level. So I think it's going in the right direction for sure.
Thank you.
Hey, Chris, it's Ann. Thanks for this. It's always good to hear the recap and hear it again and again. Is it feasible to apply the protocols and metrics to projects that are either already underway or have happened, or is this really for new projects that are being designed?
It kind of varies depending on which metric. And a lot of the suggested protocols are definitely, you know, for post-construction. And we haven't taken all of them to the level of kind of a before and after and kind of, you know, doing some sort of a statistical analysis. It's more for reporting. So for those that are already in place, some of them could easily be applied. So it doesn't have to be just kind of looking forward. And actually what we want, one of the projects that we're seeking funding for is to apply the metrics to some already completed projects to see how that all shakes out.
And Boze here again. Can I ask as a curious presenter and pop in a question?
Sure.
Chris, this is a Gulf of Mexico focused exercise. What's your read on applicability on the Atlantic and West coasts?
That's a good question. And actually that's something that we should probably try and comment on in some of our materials. I think a lot of the protocols are applicable on the East Coast, and the logic models are applicable on the East Coast. There might be a couple of because there might be a couple of outcomes that are more or less important there than the Gulf, but I think that it could be applied there. And what we try to do through this process is to document the process in a way that the Duke team is developing like a guide to how to come up with these metrics so that it can be applied in other places, but also to other restoration approaches that we didn't get to.
Any other comments or questions for Chris? Thank you very much, Chris. We really appreciate you giving us that overview. It looks like there's a lot of great resources. I'm just reviewing the website that Anne linked in the chat box, and appreciate you sharing your presentation. We'll put that up available for everybody on the box as well.
Great, thank you.
All right, so at this point, let's take a break. Let's break for 15 minutes. That will bring us back around, let's see, 5:30, around 12:40. We can get started with the group discussion on the presentations we heard today and other factors related to ecosystem services. So we'll see everybody back here at 12:40.
Thank you all.